previous two chapters, chapter 6 and 7, we discussed how to prepare the DNA from the cells and also how to evaluate the DNA quantity and quality. And now in the next two chapters, chapter 8 and chapter 9, we will discuss how to purify the protein from the sample and how to evaluate the quality and quantity of the protein. So the chapter 8, the protein preparation. So for the protein, so as we know, protein are essential cellular machinery. They can perform and enable tasks within the biological system. Each step of the cellular generation, from replication to genetic material to the cell synthesis and the death, relies on the correct function of several distinct proteins. And the precision of cellular machinery can be disrupted, and the disruption can lead to a disease. As a result, proteomic analysis has become an important technique in molecular diagnostics. The field of the field of the proteomics is the large-scale study of protein. The rapid development of the high-throughput proteomic technique has provided a valuable approach for new biomarker discovery in the post-genomic era. And the proteome is the total set of protein encoded by a genome. So that means it's the total protein complement of an organism. Many proteins are processed and modified by other proteins after translation, and the final protein complement depend on complex interaction between these proteins. Therefore, large-scale protein analysis become essential for discovering and evaluating biomarkers for personalized medicine. And then here I show the proteomics. There are two different types of the preparation for the protein that for the downstream analysis. So we can have this a peptide-based bottom-up shotgun proteomic approach. So basically, the bottom-up shotgun. So basically, you prepare you extract the protein from the sample, and then you digest protein into the small fragment of peptide, and then you separate this peptide, and so that you can analyze each individual peptide. So that's called a peptide-based bottom-up shotgun proteomics. And then also we have this top-down approach. On the left side, you can see the top-down proteomics. So you have the cell, collect cell tissue, then you extract the protein. So it's the whole intact protein are analyzed. And then that simplify the sample preparation and reduce the mixture of complexity, as no proteolytic digestion is required. And then subsequently, the specific protein of interest can be gas phase purified, and modification site can be mapped by tandem mass spec strategy. And the top-down MS provide a comprehensive sequence information for the whole protein by detecting all types of post-translational modification and sequence variants simultaneously in one spectrum without prior knowledge. So that's a top-down proteomics. Okay, that's the difference between the top-down proteomics and the bottom-up proteomics. And then the process to purify protein is, in the beginning, is very simple to the DNA preparation. You have to collect the sample, and then so you have to break open the cells, and to increase the protein concentration and purity through the centrifugation. Then you protein solubility separation using the sorting out method. And then you remove the salt, the ammonia sulfate, through the dialysis, and then you have a column chromatography based on the different property that you want to identify, so that you can isolate the specific protein. And finally, you can run the electrophoresis to evaluate your protein quality and quantity. So during the protein preparation process, then every time you can apply the centrifugation, so that you can separate your target protein from all other impurity. and then each fraction you can contain the less protein, less total protein than the beginning of the cell X-ray. But in the meantime, you enrich the target protein, okay, and that's called a cell fractionation. And the enrichment can be expressed as the specific activity. So that means the specific activity is the total amount of activity of the target protein divided by the total amount of protein in the fraction. So that's called a specific activity. So during the centrifugation, you can see so increase the speed compared to the certain fraction of the cell. Okay, so we call it a cell fractionation, and then you increase the speed every time when you try to spin down to separate the sample, the protein sample. And this slide to show the preparated centrifugation. Basically, the preparated centrifugation can be used as an initial step in the protein purification. So on the left side here, the A here from the beginning cell X-ray based on the differential centrifugation method, using the increasing you can see increasing the G force from the first step at 1,000, then 10,000, 100,000 G, the different centrifugation speed and runtime, so that it is possible to obtain at least the 4 subcellular fraction, and then each of these the fraction can be analyzed for the target protein using a biochemical assay. Okay, so for example, the cell X-ray, and then the initial spin, you can spin down the nuclear fraction, and then you remove the supernatant to the fresh tube, and then apply the higher speed 10,000 G for 10 minutes. Then you can pellet the mitochondria fraction, and then transfer the supernatant to another fresh tube, and then continue the even higher speed, the highest speed, 1,000 G for 30 minutes. Then you can spin down the membrane fraction, and finally you have a cytosol fraction. Okay, and then from the cell X-ray in the panel B here, cell X-ray through the different differential centrifugation, finally you get a cytosol fraction. Then you can analyze the specific activity. So let's say, for example, a specific activity from the beginning 20,000 activity unit over the 2,000 milligram, that's 10 unit per milligram specific activity, to the end cytosol fraction, you have 18,000 activity unit over the 500 milligram protein. So the specific activity will be 36 unit per milligram. So the purification increased 3.64. Okay, so that's the preparatory centrifugation, and then using the differential centrifugation at different time, and then to finally to purify your protein. And then of course that's the explanation to show you after the cell X-ray. But before you obtain the cell X-ray, you need to open up the cell to break the cell wall. All right, to break the cell wall. So you harvest the cell or collect the sample, and then you have to disrupt the cell, so we can have a mechanical rupture or the detergent lysis. All right, so the harsher method, you can have do the sonication. Okay, sonication, basically you have sonication, you have a cell suspension and cool on ice to avoid heating. Use the sonicator to the ultrasonic wave to break the cell, and then French press. So cells are lyzed by shear forces by forcing cell suspension through a small orifice at high pressure. Or you can use the glass bead, mortar, and pestle. Those are mechanical rupture. And then the gentle cell disruption, use the osmotic lysis. Okay. Now also you can break the cell, the osmolysis. And then also you can have a detergent lysis. Detergent lysis, so you can have SDS and ironing detergent. SDS is ironing, or we can use an ironing sodium deoxycholate or return ironing detergent, those like chops. So these can be used, all right, to break the cell. That's detergent lysis. And during the cell lysis, the compartmentalization of a cell will be partly or fully destroyed, depending on the lysis method applied. As a consequence, the hydrolysis, such as the phosphatases, glycosides, and the protease, will be present in a homogeneous protein solution and possibly alter the protein composition of the lysed cell. Therefore, enzymatic degradation must be avoided by placing the freshly disrupted sample in solution with strong denaturing agents such as 7 to 9 molar urea, 2 molar thiourea, or 2% SDS. In this environment, the enzymatic activity is often negligible. In addition, the cell disruption is often performed at a low temperature to diminish enzymatic activity. Furthermore, because enzymatic activity is pH dependent, unwanted hydrolysis can often be inhibited by lysing the protein sample at a pH above 9 using either the sodium carbonate or tris aminomethane as the buffering agent in the lysis solution. And in contrast to relatively labial phosphatase and the glycosides, some proteases are fairly resistant to denaturation, pH, and the temperature change. In these cases, it is necessary to consider the addition of protease inhibitors, because individual protease inhibitors are specific to particular class of protease. So broad range of protease inhibitors cocktail are normally used. And for example, the protease inhibitors, the people often use are PMSF or the aminoethyl benzosulfonate fluoride, tosyl lysine chloromethyl ketone, EDTA, and the peptide protease inhibitors. That's the cell lysis condition. And then you can see the denaturation agent can be an ironing detergent like sodium SDS and the cation such as ethyl trimethyl ammonia bromide. And these detergents disrupt the membrane and the denatured protein by breaking protein-protein interaction. Okay, in this category, they break the protein-protein interaction. And non-denaturing detergent can be divided into the non-ironing and return ironing. So non-ironing, you have Tridon S100 MP40 Twin 20. It's milder, less denaturing, used when critical to maintain the protein function. And then you have the return ironing, like chops. Okay, that's a detergent that you can consider. So the denaturing detergents such as SDS bind to both the membrane and non-membrane protein at a concentration below the CMC. Okay, and the reaction is equilibrium driven until it's saturated. Therefore, the free concentration of monomer determine the detergent concentration. And non-detergent detergent like a Triton S100, rigid and bulky non-polar head, do not penetrate into water-soluble protein. Consequently, they generally do not disrupt native interaction and structure of water-soluble protein and do not have cooperative binding property. The effect of the non-denaturing detergent is to associate with hydrophobia a part of membrane protein, therefore confirming miscibility to them. So in the lysis buffer, sometimes we have to add a detergent And then the detergent, there's a very important property that can it has the mechanical stimulus that can physically break apart a cell and thus allowing the chemical agent to solubilize the available protein. And that we has a property is called a critical micellar concentration, CMC. So basically, concentration of the detergent, if it's above that CMC value, then the detergent can form the micelle, and detergent additive will move directly into the micelle. The detergent with higher CMC value tend to be more easily removed by buffer exchange and dialysis, and solution with lower CMC value from the micelle require less detergent. And basically, it's more easily and generally to effectively solubilize the protein. So that's called the CMC. And then and this table just to show you the detergent with the CMC concentration. In first column is the name of detergent, and the fourth column is the CMC concentration. So you can see the tritone is 100, that's 0.24 millimolar, and the twin 80 is at 0.01. So this is an example to show the CMC value. So under the different lysis condition, then we have different quality of protein. So macro scale proteomic lysis technique can efficiently extract protein from large amount sample. And microscale proteomics focus on the separation, preparation, and the analysis of protein sample under 100 microgram, and which is very sensitive to losses in the proteomic workflow. But the issue to prepare the microscale sample is to remove the detergent contaminant presented in the lysate and avoid the loss of sample during the preparation. So the previous section we discussed how to prepare the cell to get the lysate and use different type of detergent. And so now this section we will discuss how to prepare clean protein sample for the downstream analysis. So basically, the first step to clean up the protein sample is to remove the contaminant. So you can remove the contaminant by the precipitation. So basically, use a precipitation vary based on three main factors: the detergent or the contaminant for removal, and the second, the native or denatured state of protein, and number three, the post-processing analysis. And in bottom-up mass spec-based proteomics, which investigate a peptide from digested protein, the detergent removal is critical to the bottom-up mass spec analysis. And some common detergent can be removed using acetone precipitation, and other can be removed using the TCA precipitation. And the chloroform methanol mixture or ethyl acetate can remove contaminant with high yield. And the table 6.5 here, the technique to remove contaminant and detergent from proteomic samples. So you can have the salting out, so that means you saturate the salt to precipitate the protein. Okay, and then normally use ammonia sulfate precipitation or sodium sulfate. Okay, and so the salting out. So and most of the time we use the ammonia sulfate and use ammonia sulfate, and then use high salinity. Then the high salt to precipitate the protein. Then you can remove the ammonium sulfate through the dialysis, just like this figure here. Through the dialysis, in everything inside the membrane, you have dialysis tubing, and then you have buffer, and then throughout the time at equilibrium, those small molecules, which is salt, will move out from the membrane and leaving the protein inside the dialysis tubing. Okay, so that's the dialysis to remove the salt after the salting out. And then the isoelectric point that we discussed before, so we will not spend time to discuss isoelectric point here. Okay, and we'll skip these. And then the acetone precipitation. So the sorting out, and then the acetone precipitation. So basically, you add the acetone to the aqueous sample mixture to a composition of 80%, so that you can precipitate the protein. Okay, that's the acetone precipitation, and mostly use it for the water-soluble protein. And the TCA precipitation. The TCA is specific to using the TCA is specific to a deoxycholate and a certain other detergent in the solution. So that means if you use the deoxycholate detergent, you use the TCA precipitation. And this is just to show the workflow for the TCA acetone precipitation. Okay, so for example, from the classical method, you have tissue grind in mortar and pastel in liquid nitrogen. So you have, and then you have tissue powder, transfer powder to tube, add 10% TCA acetone, free vortex, minus 20 degree for 30 minutes, then centrifuge, discard the supernatant. Okay, and then the wash with acetone, place at minus 20 for 15 minutes. So these are the protein precipitation stage, and finally you can dissolve the protein for further purification. Okay, so that's the TCA method. And this is to show you the result, the 10% TCA versus 50% acetone. And as you can see the sample on panel B here, 10% PCA, it seems more clean. After the electrophoresis, you can see the TCA 10 sample is clean. And of course, you can use the methanol chloroform precipitation. That's for the membrane protein. Okay. And the protein extraction from the archival formalin fixed paraffin embedded tissue. So protein recovery can be difficult for some fresh or frozen tissue. So when used for the proteomic analysis, the result generally cannot immediately be related to the clinical course of disease. In contrast, the FFPE tissue sample can be the alternative for diagnostic pathology archives. Use of the FFPE tissue for proteomics was traditionally thought to be too problematic given the presence of formalin-induced protein cross-link and the modification, which create a problem for separating, visualizing, and characterizing the individual protein. And furthermore, the extraction of protein from archival FFPE tissue for proteomic analysis has been hampered by the deleterious effect of formaldehyde-induced protein adduct and the cross-link that are formed during the tissue fixation and subsequent histological processing. However, recent technique development has proved that the FFPE sample represent an excellent resource. There is also the advantage of linking results with relevant clinical data, including long-term outcomes or response to treatment. And the use of FFPE tissue sample will allow sufficient number of cases of some of the rare disease or clinical subgroups to be assembled for analysis. Okay. So previous slide I mentioned about the formaldehyde can induce the adduct, protein adduct. So here is just a mechanism to show you the formaldehyde-induced modification, which can reduce the protein extraction efficiency and also lead to the misidentification of protein during the proteomic analysis. So on the right-hand side here, so you can see the protein, amino group, and then we have this formaldehyde, and then the formaldehyde can react with the N-terminal group of the lysine, cysteine, arginine, tryptophan, and the histine to form this methyl adducts. That's a 30 kilodalton extra molecular weight. Okay, and then the methyl adduct can subsequently undergo the dehydration to remove a molecule of water and to produce a Schiff base. This is a Schiff base, which is extra 12 kilodalton molecular weight. That's a Schiff base. Okay, and then you can cross-link this protein. Okay, so that's why that may cause a problem during the proteomic analysis if we use the FFPE sample. And to solve this problem, and then we can use the heat-induced antigen retrieval (HIAR) method, so that we can remove that cross-linking protein and then get the pure protein. Typically, the rehydrated FFPE section are resuspended in the 50 millimolar trace HCl pH 4 with 2% SDS, and then the sample are homogenized, followed by sonication. Subsequently, the homogenized FFPE sample are then incubated at 100 degrees Celsius for 30 minutes, followed by 80 degrees Celsius for 2 hours. And the HIAR is thought to promote high temperature cleavage of the methylene bridge and the protein denaturation, which is stabilized by selection of an appropriate buffer pH. And then you can see in the here comparison, the fray on the top, the frozen fresh tissue with the 6 molar guanidine HCl without heating. The concentration is 17.7. And then for FFP, the without heating 0.8. And then with 2% SDS with heating 6.4. And then you have 6 molar guanidine HCl with heating, then you can recover 10.9, which is very similar to the fresh sample. So that's why the heat-induced antigen retrieval method can effectively recover and purify the protein from the FFPE sample. So in this table, just to show you another example that HIAR can retrieve the sample and suitable for the proteomic analysis. This is a bottom-up mass spec analysis, so it can recover 40 to 90% overlap protein compared to the fresh tissue sample. The fresh on the top, see, you identify the protein, and then you have the formalin tissue with 6 molar guanidine HCl, and with heating from 976, you can recover A27 or A20 from formalin with heating, right? So that's the example to show the HIAR can effectively recover the protein, identify the protein. And then the next section, we will discuss the molecular separation of the protein molecules through the column chromatography. And this section basically will discuss how you target to a specific protein, specific target protein. Okay, how to isolate the specific target protein. So basically, after the cell are broken, cell extract are released, and you go through those sorting out, and the acetone or the acetone precipitation, and then recover the protein, and then protein, you resuspend the protein. And then the next step to purify protein is to go through the column chromatography. And this method uses a glass of plastic tube filled with resin, and that can separate protein based on their physical property as they flow through the column. At first, the protein sample is applied to the top of the column. As a buffer solution is flowed continuously through the column, protein in the sample migrate through the column at different rate, depending on the nature of the matrix and the physical and the chemical properties of the proteins. Therefore, the targeted protein can be separated from the other. And normally, we have these 4 different types of chromatography: size exclusion chromatography, ion exchange chromatography, affinity chromatography, hydrophobic interaction chromatography. So the next few slides we will discuss each of these specific chromatography. One is the size exclusion chromatography. So basically this chromatography separates proteins based on the size and shape on a gel filtration column, and then a smaller molecule, which means the size of the molecule smaller than the resin cutoff threshold size, will be trapped in the resin. And then the larger molecule will be excluded, therefore they can go through the resin faster and will be eluted first, and then small protein molecule will come out later. That's size exclusion chromatography. And on the right corner, upper corner here, you can see the resin, the smaller molecule be trapped in the resin, the large molecule be excluded. Therefore, the red one will go through the resin faster and be eluted first. And then the blue one be trapped inside the resin, so come out later. So that's size exclusion. Okay. Now we are going to watch the video about the size exclusion chromatography.
Separation in gel filtration chromatography is based on the differences in sizes from biomolecules as they pass through a column packed with a chromatographic medium, which is a gel. Molecules larger than the pores in the gel are unable to diffuse into the gel and are confined to the solution surrounding the beads. Molecules which range in size between the very big, excluded from the gel, and the very small can penetrate the pores to varying degrees based on their size. Access to the pores is limited by steric hindrance. If a molecule is smaller than the smallest of the pores in the gel, it will be able to enter the total pore volume.
So that's a size exclusion chromatography. And the next one is the ion exchange chromatography. Basically, they use the resin to separate protein according to their surface charge. This type of column contains a resin bearing either positively or negatively charged chemical group. Resin containing the positive charged group attract negatively charged solute and are referred to as anion exchange resin. Resin with negatively charged group are cation exchanger. In low-salt solution, protein with negative surface charge will bind more strongly to positively charged anion exchange column. Protein with a positive surface charge will bind to negatively charged cation exchange column. So later, the highest salt concentration buffer is applied to the column, so it can compete with the resin for the bound protein, and the bound protein can then be eluted through the high salt buffer. So that's the ion exchange chromatography. And then this table just to show you that the resin that we use attached for the different type of ion exchange chromatography. For the cation exchange media, so we use acidic resin. So we have polystyrene and the CM cellulose and Helix 100. You see the negative charge, negative charge, negative charge. And ion exchange media, they are the basic resin. So we have these DAE positive charge, and then we have the polystyrene resin. These two are positive charge, so they are anion exchange media. And now we are going to watch the video about the ion exchange chromatography.
Ion exchange chromatography. Proteins may have net positive or negative charge at different pH range. This is another basic rules for protein separate. If a protein has a net positive charge, it will usually bind to a column of beads containing carboxylate groups, whereas a negatively charged protein will bind to a column of beads containing diethylaminoethyl group. Protein sample loads on the column. A positively charged protein binds to the beads, and other charged protein will flow through this column and emerge first. The positively charged protein, for example, can be eluted by increasing the concentration of sodium chloride or another salt in the eluting buffer, because sodium ions compete with positively charged groups on the protein for binding to the column. Proteins with less net positive charge released first, then the higher charge density proteins. There are lots of ion exchange chromatography. You need to choose suitable chromatography depends on the character of your interested protein.
So that's the ion exchange chromatography.
Ion exchange
And then another one, affinity chromatography. Basically, this chromatography uses the principle that the protein binds to a molecule for which it has specific affinity. This is because in most instances, proteins carry out their biological activity through binding or complex formation with specific small molecule or ligand. And this small molecule can be immobilized through covalent attachment to the resin in the column. When the sample goes through the column, the protein of interest, in displaying affinity for its ligand, becomes bound and immobilize itself. The protein of interest is thus removed from the mixture of samples. Finally, the protein is dissociated or eluted from the resin by the addition of a high concentration of free ligand in solution. Because the method relies on the biological specificity of the protein of interest, it is a very efficient procedure. A typical example is the resin coupled to an antibody that recognizes a specific protein, or it may contain an unreactive analog of an enzyme substrate. So that's the affinity chromatography. And we are going to watch a video about affinity chromatography.
Separation in affinity chromatography depends upon the reversible adsorption of biomolecules through biospecific interactions on the ligand. Most affinity chromatography experiments are performed in three main stages: equilibration, sample application and wash, and elution. The first step is the equilibration of the stationary phase to the desired start conditions. The second step is sample application and wash. The goal in this step is to bind the target molecules and wash out all unbound material. Non-binding molecules will pass through in the flow-through during washing with binding buffer. In the third step, elution, biomolecules are released from the biospecific ligand into the elution buffer by change in the buffer composition. A common way is to increase pH of the buffer.
So that's the affinity chromatography. And then the last one is the hydrophobic interaction. So in this purification, protein pass through chromatography column packed with a supported resin to which the hydrophobic group are covalently linked. Phenylsulfurose, which contain a phenyl group, is commonly used in this column. So normally the protein sample is prepared in high salt. So when you treat the sample with high salt, then those hydrophobic amino acid residue will stick outside, will be outside, exposed that the hydrophobic interaction, hydrophobic residue. Then when you go through the column, then the protein will bind to the phenyl group through the hydrophobic interaction. Okay, when the sample goes to the column, and then later on when you remove the salt, then the protein conformation changes back to the original conformation, and so that they will be removed from the hydrophobic interaction. So that's how you purify the protein through the hydrophobic interaction. So here's the schematic diagram of the HPLC system, the high pressure liquid chromatography. So that you have this HPLC solvent from left to right and HPLC pump. You have injectors, the sample inject to the system, then the sample goes through the column. So the column, based on those previous discussed different type of chromatography, affinity chromatography, ion exchange chromatography, hydrophobic interaction chromatography, and the size exclusion chromatography. So they can separate the protein. So the principle of the column is the same. Just you have these high pressure liquid chromatography to push the sample through the column. So that's HPLC. So in the end of the protein purification, then you want to calculate the purification efficiency of a target protein. So that you have to familiar yourself with this table. So the first column here, the total protein, they say the first one, from the beginning, the crude cell extract, you have 2,000 milligram protein, okay? And then you have the subcellular fraction using differential centrifugation is 500 milligram. So, and then the total unit activity from 20,000 and then down to 18,000. All right, so that's the first step, the subcellular fractionation using the differential centrifugation. So the total protein down from 2,000 to 5,000, and total activity down from 20,000 to 18,000. So the specific activity, okay, basically would be 20,000 divided 2,000 is 10 here, and 18,000 divided 500 is 36. So the full change, the impurity will be 36 divided by 10, you get 3.6. So that's 3.64 purification. And the yield, okay, the yield is 18,000 divided by 20,000, so you get 90%, all right, 90%. And total multifold purification is 3.6, so 36 divided by 10, 3.6. And then you move on to the next step. After the ammonium sulfate precipitation, total protein down to 100 milligram, and then activity down to 16,000, right? And so the specific activity will be 16,000 divided by 180, get you 89. And then the 89 divided by 36, increased purity is 2.5, okay, 2.5. From the subcellular differential centrifugation to ammonia sulfate precipitation at 2.54. And the total yield is 80%. So it's 16,000 divided by 20,000, 80% total yield. And total multifold purification is 89 divided by 10, so 8.9. And then if you further down to the gel filtration chromatography, so from 180 to 45, 16,000 to 13,000, then the specific activity will be 13,000 divided by 45 is 289, All right, 289 divided by 89 is 3.2. And then the 13,000 divided by 20,000 is 65. And then 289 divided by 10, so it's 228.9. So that's how you calculate the purification efficiency of target protein after each step of purification. So I hope you will familiar yourself with this table and how to do the calculation. So let's say for the chapter 8,